So, thanks very much indeed for inviting me to speak to you. I'm, for the first part, going to talk about BBSRC's perspective on this, wearing my BBSRC hat. For folk who are not from the UK, I'm on secondment from being an academic helping out with one of our funding agencies, which is the BBSRC. And then I'm going to talk a little bit about a couple of studies I've been involved in with my academic hat that are actually genuine synthetic biology, which is what that says there. So there is indeed a definition. Like all definitions, it is somewhat elastic. I think like an elephant, it's easy to identify when you see it, but less easy to define and describe, and we're better off with that perspective. But there's a recognition from most people that, in contrast, say, to metabolic engineering, it does actually normally involve starting with something quite fundamental, probably at the DNA level, rather than cloning something that's already there and PCRing it and things like that. It is a recognition that some of the principles of engineering that I'm sure Richard will talk about shortly are important to the whole process, the recognition of modularity and the like, which have actually been important in real evolution too. And I think it's recognised that the reason most people are interested in this stuff is not because it's telling you about existing biology, but it's going to help you use biology to make exciting things, whether as sensors or industrial biotechnology products for sustainability, for when, or preferably long before, the oil runs out, we realise we've got to make things sustainably, and we're largely going to be using biology to do that, starting with carbohydrates rather than hydrocarbons. It covers a whole multitude of spaces. Again, I'm not going to talk in great detail about that at different levels of engineering. It certainly includes metabolic engineering, and I wouldn't say otherwise. Appropriate enough in this Centre for Nanoscience, it's recognised that it probably includes nanoscience and nanotechnology. It certainly includes systems biology and the modelling, so that you go into making something with some idea of how it ought to behave once you have made it. And it certainly exploits the fact of next generation sequencing. The amount of bases that was sequenced by Fred Sanger in 1977 for his first paper was 5,000. It took him a year and a half. One machine now does that in a millisecond. So that's where we've come, and in so many years' time, maybe SynBio will have gone to a similar kind of place. The argument has been made that it is really about recognising that biology is really just like circuits and electronic circuits, and how you put them together into chips and computers and so forth at different levels of organisation. It's not an unreasonable analogy. I don't have any problems with it at all. Some people may find it helpful, especially if they come from an electrical engineering background. And there's a recognition that I've nicked from one of Deck's papers here, that you can consider things in terms of two orthogonal sets of properties: the complexity on the ordinate of the components, and on the abscissa, how similar to or different from natural things, the things you're wishing to play with are, some of which may be completely abiological, and some of which may be actually just a little bit of biology that's been tweaked. And I think that's a very reasonable way of looking at the kind of landscape in which we are interested. Folk, and I think largely in the UK that means BBSRC and EPSRC, with some money from ESRC and AHRC, have actually been funding SynBio for quite some time, including through response mode well before 2006, and I'll give an example of that later. There's a whole bunch of stuff there which you can't possibly read or would wish to in the time available. I've put three in bold that I will just mention briefly as examples of some of the activities that have been done. The main message is really, as if there was any doubt, research councils and indeed the science minister recognises absolutely the importance of SynBio. When I speak with him, I say to him, well, the point about this, of course, is with SynBio we can make anything. Now you have to translate into what that actually means and what you want to make, and you're limited only by your imagination, really, not by the likely ability of biology to be able to make whatever it is that you are interested in. So I'll talk briefly about the networks in SynBio that were quite early on in the piece where we put this together. I'll talk very briefly about the things we're doing with the NSF on enhancing photosynthesis, and we've recently had a joint synthetic biology initiative with MRC, EPSRC and the DSTL. Networks in synthetic biology, 4 got funded in different parts of the space, written there, and you can see the funders at the bottom. That was all about building communities. It's a typical way we go about things when something is moderately new. You don't necessarily know who all the right players are, but you want to get them together to develop the best kind of thinking, and so you do that by building capacity through networks, and that was exactly what was done there. So I think that's been very successful because we have built quite a lot of capacity in a fairly short time, as Anne's pictures from the Oldham paper just showed. Going back to 2010, our own strategic plan on the left, we've long recognised that this is going to be important, particularly probably in the theme that we have on industrial biotechnology and bioenergy, making microbes and other organisms, plants, particular focus of this meeting, do stuff, and that's down as Grand Challenge 2 in the top right of this, and a recognition that the amounts of data that are available of all kinds that must be turned into knowledge, whilst not of itself biology, is an important contributor to biology, and that must be funded as well, and we're very keen on that. At the moment, we've got an investment of about 38 million in SynBio, or things badged as SynBio. There are, I would say, plenty of things that are SynBio that weren't called that when they originated, and so that's probably a lower estimate. We try where possible to fund stuff through responsive mode. Sometimes we have to have calls and highlights just to point people in the right direction, which is directed. And we also have, mentioned at the bottom, contributions to an ERANET, a European research area network, in which lots of countries come together but spend money on their own countries. For synthetic biology is about $1.5 million of our money into that. And again, as it says there, lots of areas of application that you might use it for. We've been promoting our so-called strategic longer and larger grants, LOLAs, particularly for white or industrial biotechnology, but also there's been one awarded in an underpinning space with using click chemistry to make DNA in a perhaps more efficient way than phosphoramidite chemistry historically allows you to do. There's also been a European scheme, Eurocause, which we've invested in as well, and I've mentioned the Joint Synthetic Biology Initiative, which was very well subscribed, and so it's encouraging that people are wanting to use these things for, mainly in this case, diagnostics and sensing. And finally, we had, based on a sandpit, an exciting joint program with the National Science Foundation seeking to enhance photosynthesis, since whatever we do, whether it's improving crop yields for foods or improving crop yields for non-foods, for fixing carbon, we are going to need to improve, by definition, the yield of photosynthesis, because we aren't probably going to affect the number of photons coming in, but we can affect the efficiency with which they do something. And finally, as Anne also mentioned, there is purposely, recognisably, and importantly, a major job to be had involving the ethics and public dialogue of synthetic biology. And that has been going on for quite some time. With EPSRC, we've had a synthetic biology dialogue very successfully for quite some time, quite structured, and all of that's been published, and all of that is leading to a so-called UK roadmap on SynBio that is going to be published fairly shortly. And there's an expectation that the Minister of Science is likely to call for a leadership council in this space, although that's up to him. The Enhancing Photosynthesis Ideas Lab, so-called, or sandpit if you prefer that terminology, has funded 4 quite large projects, and in the plant space we've also got Synthetic Plant Products for Industry as one of those synthetic networks, and a recognition in the plant space that SynBio is going to offer tremendous opportunities in all kinds of areas. I don't really wish to limit it by even defining them. There's a few listed there. It's up to the imagination of researchers to decide what it is we want to be able to do. On Anne's slide there was a picture which probably was of this vaccine that Polly Roy has produced using synthetic biology. It's an example of one particular product that once you realise you can make anything and lots of variants thereof, we still don't understand what makes a good immunogen on a vaccine, and SynBio is going to allow us to make lots and lots of different ones and work out which are going to be good ones and get much more quickly than historically. It takes about 1.5 years to make a vaccine for some emerging pathogens such as flu, which seems absurd. They're still made in chicken eggs. I mean, come on. So I think we've got way to go here before we really can ramp things up, but the gains to be had are absolutely enormous. And then folk will be well aware from quite a lot of press recently of a trial of a wheat crop at Rothamsted Research, genetically modified to produce a sesquiterpene, which is an aphid alarm pheromone, which makes the aphids think that it's not a nice place to be. It's a natural product produced by all sorts of plants exactly to this purpose, and what John Pickett and his chums have done is put it into wheat, and they will in due time see whether or not this decreases the amount of aphid attack, and obviously that's important to our food security agenda. In microbes, I think most people recognise that's probably the major area where we're going to be able to do things for all kinds of reasons connected with GM legislation in Europe. And there's a list of activities there. Biobutanol production funded a number of places, and riboswitches, which I suspect is Jason Micklefield's project in Manchester. So lots and lots of different kinds of things that we can do to improve the way in which we get bugs to make drugs. That's what it comes down to. And again, a few more listed things there. Some of the stuff that Jason Chin's been doing at the Laboratory of Molecular Biology, more than 21 amino acids, a tetranucleotide genetic code, and all of these things give one the kind of control over microbial performance that we've never previously had in the era of metabolic engineering and the development of modern fermentation, post the ability to do genetic engineering, as it was called when it first came in. And finally, I'm going to give a couple of examples of real synthetic biology. The first is a project that was started by Simon Gaskell and Rob Bainan, Simon in Manchester, Rob in Liverpool, to develop a method that would give you absolute measurements of protein activity. Classical isotope dilution-based methods give you relative performances or relative expressions in proteomics experiments, but this is an absolute method. And the way it works is as follows. You work out, top left, the proteins you are interested in, and you find a peptide that is unique in mass to those proteins, and you can do that by sequence gazing, and there are algorithms for doing that. And then you make synthetically, hence the SynBio thing, in this case E. coli, a concatenated version of these signature peptides, or the genes encoding said signature peptides, which end with a lysine or an arginine, so they can be subject to tryptic digestion. And you can then grow them in E. coli, isotope labeled again with heavy arginine or heavy lysine, and extract the Qconcat, which has tags, his tags at the end and so forth, and weigh it. And that's where you're going to get the absolute quantification from, because you've actually weighed the thing. Then you add it to the sample in which you are interested in, add your trypsin, the tryptic peptides come out. Some of them are heavy from your Qconcat, some of them are light from the natural organism, and obviously by ratioing the two, and it's pretty obvious here. I mean, the mass specs are a bit too small to read, I expect, and I'm probably going to do something dreadful if I do that. But it's pretty obvious that one is displaced by either 6 or 12 mass units from the other, and by simply ratioing the mass spectra in the identical conditions, you get an absolute expression value for the amount of protein in a cell of interest. And with Rob and Simon and a whole bunch of other people based in Manchester, we did exactly that for, in this case, on this published paper, all the glycolytic enzymes of yeast. And so the sort of result that comes out is an absolute measure of the number of molecules per cell of each of, in this case, the glycolytic, and you can see some other enzymes in there from these Qconcats. There were about 100 different signature peptides in that particular Qconcat, and this kind of information is absolutely required for constructing systems biology models that tell you the amount of protein. Then you may know it's Km and it's Kcat and so forth, but you need, for the Vmax, the amount of for each protein that is there. That sort of method allows you to do that. My second example is based on aptamers. Now, the traditional method for devising an aptamer is a nucleic acid sequence that binds, or it has a fit, apt, Latin, to some target molecule, was to make a whole bunch, 10 to the 14 or 15, random oligomers, oligonucleotides. Then you could pour them down a column with your target on it, and some of them would stick. You could wash them off, PCR them up in a non-accurate way to get mutant versions of the nucleotides that had bound, and do that again round and round and round. And at the end, you might sequence the thing you'd made just to see what it was out of interest. But at no point in the intermediate steps did you know what you had made, because you just made it by sloppy PCR of whatever you'd happened to have bound the first time round. And obviously that's not very effective, and you would typically get 1 out of it, but 1 out of 10 to the 15 isn't kind of not that very efficient. If you've read the abstract, all of this is a combinatorial optimization problem. And what we did was essentially to recognize, CLADE stands for closed loop aptameric directed evolution, that if you actually knew what you'd made at every step of the way, you could do this in a much more efficient and effective manner. And that's essentially what we did and do. You start by making some sequences directly in a synthesizing machine. In this case, it was a machine made by Combinatorial that does it all on a dial-it-in kind of Affy chip based on electrochemistry and phosphoramidite chemistry to make these sequences you decided based on whatever you like, random background knowledge, any kind of things. We had reasons for choosing things. And then you go and make the measurement. In this case, the measurement was, would it bind APC, alpha-ficocyanin, a fluorescent target? And after a while, you can see that it's got some range of binding, but you know the sequence at every position in the aptamer array. So you've got automatically, in this case about 5,000 it was in the early stages of this, sequence activity pairs for each generation. Then having worked out what you've got, you can then make another lot based on the knowledge you've acquired as to which ones were good and which ones were no good. And then you go round and round the loop until you finally got there. And when you look at the data, you can see in the first generation, they're rubbish. You hardly get any that bind very well. These are the medians in the middle. And by the 9th generation, you've actually got one that binds really, really well. But instead of having to do 10 to the 14 or 10 to the 15, we did 4 times 10 to the 4. 9.5 orders of magnitude more effective. The search space, these are 30mers, it says in the abstract, is 10 to the 18, and you're not going to go there in any realistic timescale. Also quite interesting to see the comparative binding to DS-Red, and there are some that actually bind DS-Red quite well, but mostly they don't. And if you had an interest in doing evolution in parallel, there isn't any particular reason why you couldn't do all of that. Again, the principle is the same. You can do what you like because you know all the sequences and you know all the fitnesses, and that allows you to do the evolution with your eyes open. And when you do that, you begin to understand the landscape of binding. That's a particular way of describing it, where there are 2 major basins of attraction of different things. And in fact, those were 30mers. And then the cute thing we could do was to learn the relationship between the sequence and the binding affinity by training a mathematical model. In this case, we used a technique called random forest. Details don't matter. Standard machine learning approach has some advantages. And we could then learn the model and throw 10 to the 6 in silico sequences at the model and then say which ones look good. So we don't have to do the 10 to the 6, we can choose which one we do. We actually chose ones predicted to be up the curve, and the correlation coefficient, as it says in the abstract book, or in the paper anyway, was about 0.87. So we'd learnt the relationship between the sequences and the fitnesses for all sequences, even huge out of sample things. Actually, the ones that we did in the test set here, which is an independent test set, actually bore very little sequence relationships to the ones we'd learnt in the first place. So these machine learning models are very good at capturing the knowledge underpinning the thing. And then finally, we actually did all 10mers rather than 30mers, which is just over 1 million, 2 to the 10. And that allowed us to understand completely the landscape of sequence versus affinity. And that allows you to do interesting things if you're interested in what landscapes of this type look like, and for those who are into this kind of thing. It turned out to be quite similar to an NK landscape with K just shy of 1. And so that allows one to begin to understand, because we've got, by synthesis, by synbio, the sequence and the fitness for, in this case, 1 million of these things, the whole landscape of interest. Conclusions. We, as many other RCs, are now very much of the view that this is an extremely important area for all sorts of intellectual and economic benefit reasons, and the Minister of Science has said so on many occasions himself. There's already a bunch of stuff being funded, in our case £38 million worth, and I do stress that really in synbio we can make anything. The potential is limited only by our collective imagination. But we do have to recognise that anything you make as a sequence, the number of sequences scales exponentially with the number of positions in those sequences, so it's a combinatorial optimisation problem. And finally, I gave you a couple of examples based on Qconcats and Clade, both of which are genuine synbio approaches based on synthesising nucleic acids in a known, determined, engineered way. And with that, I will thank you for your kind attention.